Welcome everyone. Today we are going to learn about asymmetric distribution of lipids in the plasma membrane. This is an important property of plasma membrane, but before I begin, I want to mention three important facts about the plasma membrane. So the first point we have to remember that I have already discussed in my previous video, basic facts on plasma membrane, that membranes are lipid bilayer in nature. Lipid bilayer. This is the first point we have to remember. Then we can understand asymmetric distributions. Then second point is topological equivalence. Topological equivalence. And the third is classification of lipid molecules. Classification of lipids in the plasma membrane or in the membrane. I have made separate videos on all these topics, but I will also explain here briefly. Let me draw an eukaryotic cell, then we can all understand these 3 points. Let's suppose this is an eukaryotic cell. We know plasma membrane surrounds the boundary of a cell. So let me draw this plasma membrane in green color. Now, why I have taken an example of eukaryotic cell? Because eukaryotic cells contain intracellular organelles like nucleus, endoplasmic reticulum, Golgi bodies, then lysosome, peroxisome, endosome, mitochondria, chloroplast. Okay. And in my previous video, I have already mentioned basic facts that these intracellular organelles are also surrounded by membranes. So let me draw in green color. So that's why I have not taken example of prokaryotic cell, because prokaryotic cell don't have any intracellular organelles. So I have taken the example of eukaryotic cells. Now, if we see all these membranes with the help of electron microscopy, then you will find that all the membranes are actually lipid bilayer. Now, if it is lipid bilayer, let me draw another layer of the lipid. Now, let me draw the outer layer of the plasma membrane in red color, and let's suppose this green color is the inner layer. So let me draw the lipid bilayer. So if we see these membranes with the help of electron microscopy, then we can actually understand that this is lipid bilayer. So both these green and red layer, lipid layer, makes the plasma membrane. Okay. Now, in case of plasma membrane, the red or outer layer of the membrane is called exoplasmic leaflet. Exo means outer. Exoplasmic leaflet. And this inner layer is called cytosolic leaflet. Now, why it is called cytosolic leaflet? Because this green layer of the lipid faces the cytosol, and the outer layer is actually called exo, means outer. Now, this is the first point we have understand, that if we see all these, if we see plasma membrane with the help of electron microscopy, we will find 2 layers. But I have mentioned that all the membranes are lipid bilayer, so there will be another lipid layer in case of intracellular organelles. Now, here comes the topological equivalence. In case of plasma membrane, the layer that is outside called exoplasmic leaflet. In case of intracellular organelles, that layer will face the lumen of the intracellular organelles. So lumen is called here. This is the lumen of the intracellular organelles. These are the lumen. So now here, in case of intracellular organelles, the outer layer will face the lumen of these organelles. This is called topological equivalence. Topology means positional, equivalence means equality or same. As because we can also, obviously I have made a separate video, I am not talking in details, but you can watch the video also if you want to know more about topological equivalence. As you see that this is the cytosol, okay. So here this layer facing the cytosol, that is why we call cytosolic leaflet. Also for intracellular organelles, this outer layer will face the cytosol. So the composition of lipids should be like cytosolic leaflet. So now our outer layer of this intercellular membranes, actually outer lipid layer will actually the cytosolic leaflet, and the inner layer is actually the exoplasmic leaflet. In case of plasma membrane we call this, but in case of intracellular organelles we call the luminal phase. We call the exoplasmic leaflet luminal phase of the lipid. Luminal phase of lipids in case of intracellular organelles. Also for intracellular organelles, the cytosolic leaflet we will call cytosolic phase of the lipid, cytosolic phase. So the name of for cytosolic is same for intracellular organelles. So for intracellular organelles we will call luminal phase for the exoplasmic leaflet, and for cytosolic leaflet that is cytosolic leaflet because it faces the cytosol. So for intracellular organelles. Now if you want to understand this topological equivalence, you have to understand the process called endocytosis. I am not explaining here this topic. Now in case of classification of lipids, there is three major category of lipids present in the plasma membrane. One is called phosphoglycerides. Another is called glycerides, and this is called sphingolipids. And another is called cholesterol. Under the phosphoglycerides, there is major phosphoglycerides like phosphatidylcholine, phosphatidylinositol, phosphatidylserine, phosphatidylethanolamin, etc. Under the sphingolipids, there is sphingomyelin. I have already made separate video on this, so I am just briefing all these points. There is sphingomyelin, there is cerebrocyte, and there is gangliosides. Okay, and there is cholesterol. Another way of classification of lipids is phospholipids. Phospholipids, glycolipids, and cholesterol. Okay, so here phospholipids means those lipids contain phosphate like phosphatidylcholine, ethanolamine, inositol, serine. All these phosphoglycerides contain phosphate, that's why they fall under phospholipids. But in case of sphingolipids, only sphingomyelin contains phosphate group, so it is also fall under phospholipids. So all this falls and only sphingolipids. Now cerebrocyte and gangliosides contain sugar molecules, so glyco means sugar, so these two lipid molecules also fall under glycolipids. It has been found that although most phospholipids are present in the both lipid bilayer or lipid layer, but some are more abundant in one particular layer. That's why it's called asymmetric distribution. They are not distributed symmetrically. Now let me give example. Scientists observe this phenomena on two important cells. One is red blood cells and another is called on red blood cell and MDCK cell line, Madin-Derby canine kidney cell line. So they observe the lipid composition of plasma membrane in these two cells, and they found that almost all sphingomyelin and phosphatidylcholine, these two lipid molecules are present in the exoplasmic leaflet. Exoplasmic leaflet. They observe another point, that is phosphatidylethanolamine, phosphatidylserine, and phosphatidylinositol. These three phosphoglycerides are preferentially present in the cytosolic leaflet. So that's why I have already discussed about what is exoplasmic and what is cytosolic leaflets. So it will help you to understand the asymmetric distribution of lipid molecules. So these three phosphoglycerides are preferentially present in the cytosolic leaflet. They also observe that Glycolipids, that means those lipids contain sugar molecules. Glycolipids are exclusively, remember this, exclusively means there is, you will not find otherwise. So exclusively present in the exoplasmic leaflet. So there is no glycolipids in the cytosolic leaflet. So all glycolipids, okay, almost, not almost all, but all glycolipids present in the exoplasmic leaflet. Okay. Another point, they discovered that cholesterol are actually present or actually distributed evenly. That means they do not fall under asymmetric distribution of lipid molecules. So they are evenly distributed. That means 50% cholesterol present in the exoplasmic, 50% present in the cytosolic. So there is no asymmetry in case of cholesterol. So we will remove cholesterol from this list because they are present evenly. Okay, so these lipid molecules are actually showing asymmetric distributions. Now, how scientists understand that or know this asymmetric distribution of lipid molecules? They actually use one enzyme. I have already also made a separate video on the enzyme called, they have used one enzyme or take the help of one enzyme called phospholipase. Now, to understand how phospholipase works, actually I have to draw one phosphoglycerides. I have made separate video on phospholipase. So in this video I am also going to show you, then you can understand. Let's suppose I am giving one example of phosphoglycerides. In case of phosphoglycerides, there is glycerol. There is glycerol molecule present. This is the backbone of the molecule, and glycerol is very simple molecule because it contains only 3 carbon. With the first carbon there is one fatty acid is attached. With the second carbon there is another fatty acid attached. With the third carbon, one phosphate group is present or attached, PO4 minus this group, and with the phosphate group, one head group is actually attached. Okay, remember this. Now we know that this head group and phosphates makes the polar head group. And this polar head group is actually hydrophilic in nature, and this portion, that the fatty acid, so these are fatty acid chain, these chains actually make the non-polar portion of the lipid molecule, or non-polar portion of the phosphoglycerides, or we can say this is hydrophobic in nature. Hydrophobic. So this is an example of a phosphoglycerides. Any phosphoglyceride it could be. Phosphoglycerides. Now, depending on the head group, we call this phosphatidylcholine or ethanolamine or serine or inositol. Now, phospholipase enzyme, you can say there is 4 phospholipase enzyme can cleave this molecule in different positions. Like phospholipase A1, this enzyme can cleave the bond between first carbon and first fatty acids. Phospholipase 2, PLA2, can cleave the bond between second carbon and the second fatty acid. Phospholipase, now this is not A3 but phospholipase C, can cleave the bond between third and phosphate group, and phospholipase D, this enzyme can cleave the bond between phosphate and the head group. So this phospholipase enzyme can cleave the phosphoglycerides. Now scientists taken the help of these 2 enzymes, phospholipase C or phospholipase D, any of the enzyme, not with this enzyme. So with the help of phospholipase C or phospholipase D, we can cleave the head group. And I have mentioned that the head group actually differs, because the head group could be choline, it could be ethanolamine, it could be inositol or serine. So basically this structure is same for all the phosphoglycerides like phosphatidylcholine, phosphatidylserine, phosphatidylinositol, and phosphatidylserine and phosphatidylethanolamine. So all these 4 different phosphoglycerides, this structure is basically same, but head group differs. To identify which molecules is present, so we have to take this help of these 2 enzymes, because with the help of PLC, it can cleave the phosphate and head group. Then we can chemically analyze the head group. Also, we can use PLD, then we will only find the head group and we can analyze it. So scientists use these two enzymes. Now, they have taken this PLC or PLD, these two enzymes, and they digest cells. Now, this enzyme cannot penetrate inside. That's why if this is the enzyme, black color, let's suppose this is the enzyme, then it will only cleave or digest or hydrolyze the lipid molecules present in the exoplasmic leaflet. It cannot hydrolyze the inner side. It will hydrolyze the outside of the leaflet. Then they observed chemically what kind of molecules present outside, and they found one interesting observation that proves that there is asymmetric distributions. So let me explain. Now they find that, let me draw one graph. Let's suppose this is the exoplasmic phase of the membrane and this is the cytosolic phase of the membrane. They find that approximate 80%, let's suppose this is the 100%, this is 80%. They found that approximate 80% of the phosphatidylcholine is digested with the help of PLC and PLD. What does it mean? That it means that among the 100% of the phosphatidylcholine, 80% is present outside, because 80% of the phosphatidylcholine is hydrolyzed in the membrane. They find that only 20% of, let's suppose this is 20%. So what will happen? Phosphatidylcholine is present just 20% inside. So here only 20%, okay, in the cytosolic leaflet. But in case of phosphatidyl ethanolamine, they find that only 20% is hydrolyzed. That means it is present in the exoplasmic leaflet, because this enzyme cannot enter inside the cell. So it will only cleave or hydrolyze the outer layer, and this is the result of the outer layer. So that means there is 80% P is inside. And they also found that less than 10%, let's suppose if this is the 10% of the lipid, then less than 10% of the lipid is actually phosphatidylserine in the outside. So more than 90% present inside. As you see, I have mentioned that PE, PS and also PI. PI is very actually present in low amount in both bilayers. So, but also it is present inside more. So these three PS, PE, PS and PI, these three lipid molecules or phospholipids present less amount, approximate here 20%, approximate or less than 10%, and this is also same, and here is approximate 80%. So from this observation, they found that these lipid molecules are not symmetrically distributed. That means they are not present 50-50%. They are asymmetrically distributed. And also they found that sphingomyelin is present more percentage than PC. That means approximately 90% of sphingomyelin and 10% only inside. So this is for sphingomyelin, okay, approximate 90% present outside, and the rest is the inside or cytosolic leaflet. In case of glycolipids, 100% of all glycolipids, that means cerebroside or gangliosides, are present. So these are glycolipids that are all present in the outer layer. But in case of cholesterol, they found that this is 50-50. That means 50% present outside, 50% present inside. So this is for cholesterol. So from this experiment, they found that there is asymmetric distribution of the lipid molecules. Now, so this is 50%. This is 100% glycolipids. And this is 50%, 50%. So this is glycolipids. Now, as we know that from one answer, in case of science, from one answer, more questions arise. Now we have got that these molecules or these lipid molecules are asymmetrically distributed. Okay, fine. Now where from all this asymmetry is generated? where from these molecules are actually that means that why from this asymmetry is generated. And scientists actually pointed that the position where these all molecules or lipids are synthesized may be the answer. Why I am telling it? Because in case of sphingomyelin, as I have mentioned under the sphingolipids, that sphingomyelin, cerebrocyte and gangliosides, that means all sphingolipids are actually ultimately generated in the Golgi apparatus. So all the sphingolipids, it could be sphingomyelin, it could be cerebrocyte or gangliosides, that all made in the Golgi body or Golgi complex. Now, the enzyme that adds the head group, and the head group actually determines the nature of these lipid molecules, because the basic structure is same, but the head group is different. Now the head group is added in the Golgi complex, and this enzyme that actually add the head group is present in the luminal phase. Let's suppose this enzyme actually adding the head group, and this is actually facing the lumen, or this enzyme is present in the lumen. So this lipid molecules are actually present in the luminal phase. I have written here the luminal phase, that means the red color lipid molecules, okay, that I am denoting as red color. Now when membrane lipids are synthesized in any lipid layer, they are actually distributed to the plasma membrane also. From the Golgi complex, these lipid molecules will actually then go to the plasma membrane. From the endoplasmic reticulum, basically all phosphoglycerides, I'm writing here, all phosphoglycerides like phosphatidylcholine, phosphatidylinositol, phosphatidylethylamine, phosphatidylserine, all phosphoglycerides are synthesized in the endoplasmic reticulum. Remember this. But here, the enzyme that actually adds the head group present in the cytosol. Now, in case of endoplasmic reticulum, all the phosphoglycerides that are generated, the enzyme that adds the head group present in the cytosol. So that means all these lipid molecules will present in the green color. So when these green lipid molecules are delivered, it will go to the green layer of the lipid molecules. That means cytosolic leaflet. It will go to the cytosolic leaflet. That's why I have mentioned the second important point, topological equivalence. Scientists observe that these molecules like sphingomyelin, glycolipids, they are generated in the Golgi, lumen of the Golgi, and that's why they are present exoplasmic leaflet. They are present more in the exoplasmic leaflet. And all these phosphatidylcholine, inositol, ethylamine, and serine, all these molecules are actually generated in the endoplasmic reticulum, and the enzyme that add the head group is present in the cytosol. That's why it is present, this lipid is present in the cytosolic leaflet. So it will go to the plasma membrane also, and all lipids are present in the cytosolic leaflet of the plasma membrane. This is the topological equivalence. Now one questions will actually come. Yes, for phosphatidylinositol, this is present inside a cytosolic leaflet, okay, more. Phosphatidyserine is also, inositol is also. But what will be the answer for the phosphatidylcholine, because it is present outside or exoplasmic leaflet more than 80%. Now here comes another questions, and scientists discovered another time that there are some molecules or proteins present that is called translocator. This translocator actually can help in the performance of one movement of the lipid molecules, that is called flip-flop movement. Now what is flip-flop movement? I have mentioned in my previous video that if lipid molecules jump from or migrate from exoplasmic leaflet to the cytosolic leaflet, or from cytosolic to the exoplasmic leaflet, this is called flip-flop movement. So basically scientists told that phosphatidylcholine are present in the cytosolic leaflet, but one translocator actually helping these lipid molecules from migration from cytosolic to the exoplasmic. That's why they are present more in the cytosolic leaflet. Now, what are the translocator that helps in this type of movement? Let me write here. Now, there is three type of translocator proteins are present. One is called flippage. This enzyme, this basically their enzyme, this enzyme can transfer or migrate lipid molecules from exoplasmic to the cytosolic. Another type of enzyme is present called floppage. They can transfer the lipid molecule from cytosolic to the exoplasmic leaflet. And now there is another group of enzyme present that is called scramblage that can move the lipid molecules from either side, from exoplasmic leaflet to the cytosolic or cytosolic to the exoplasmic, in both directions. Now in case of phosphatidylcholine, this floppage enzyme will work because here the lipids present in the cytosolic leaflet, but it goes to the exoplasmic leaflet. So this is the main reason of the migration of or presence of the phosphatidylcholine in the mode in the exoplasmic leaflet. Now, is there any benefit of this asymmetric distribution of lipid molecules? Yes, there are some examples of property that are represented by the plasma membrane, like the presence of phosphatidylethanolamin inside in the cytosolic leaflet helps to make curvature in the plasma membrane. Curvature. Okay, I have made separate video on this, how lipid molecules actually helps or lipid composition helps in the properties of plasma membrane. So presence of phosphatidylethanolamin helps in curvature, and curvature occurs during viral budding, then cell fusions or movement, cell movement or locomotions. So PE that is present more cytosolic leaflet helps in the curvature. Then another point we can remember, that is presence of phosphatidylserine inside, and during apoptosis this phosphatidylserine actually move from cytosolic leaflet to the exoplasmic leaflet because presence of this floppage enzyme. Yes, floppage enzyme. This enzyme helps to migrate or flip-flop this phosphatidylserine from cytosolic leaflet to the exoplasmic leaflet. Now, in normal cells, phosphatidylserine is not present in the exoplasmic leaflet. Very low amount, less than 10%. So when the amount of phosphatidylcholine increases, more than 10, 20, 30% increases, then macrophage and phagocytic cells in the immune system can identify that there is some problems in the inside the cells. So this is apoptosis. During apoptosis, phosphatidylserine comes in outside exoplasmic leaflet, and scientists can also detect the apoptosis by the help of one protein called annexin 5. I have mentioned already. And if you tag these annexin 5 proteins with fluorescent dye, then you can also identify which cells are going apoptosis by flow cytometry. So this is another point is also for phosphatidylserine, that during blood clotting in the initial stages in the platelet cells, this phosphatidylserine also migrates from cytosolic leaflet to the exoplasmic leaflet, so to activate clotting enzyme. Hope this video will help you. Thank you for watching, and always keep learning.